Department at the University of Trieste in Italy. He started his postdoctoral studies in molecular pathology department on the International Center for Genetic Engineering and Biotechnology in Trieste. At present, Emmanuel Burati works as contracted professor on the Department of Life Science in the University of Trieste. He is also the principal investigator of the Molecular Pathology Research Group in the International Center for Genetic Engineering and Biotechnology. His research is implicated on studies of RNA metabolites, especially the pathological effects of pre-mRNA splicing in human disease. More specifically, his lab is investigating the role played by nuclear factor TDP43 in ALS and FT, lobar degeneration, and the rescue of normal GAA splicing profile to function as a potential therapy for late onset bone disease. So, Emmanuel, when you want, please.
Thank you very much. Thank you, Javier, for having organized this wonderful workshop. What I'm going to tell you is some of the latest results we have had on the search for TDP43 modifiers. And the reason why we are looking for TDP43 modifiers are basically two. One of them is that we have already seen this morning for quite a few speakers that ALS is a very variable disease. So I think the implication of this is that there are many modifiers around the disease. And secondly, with my okay, and secondly, I would say that I'll always like to remind the people that TDP43 is not a brain-specific protein, right? It's a protein that can be found all over in our body. It's completely essential for the development of the fetus. If you get rid of TDP43, any cell that you can think of is going to die. And so, in some ways, also TDP43 is not linked to the ALS FTLD spectrum of disease, because now we know that aggregates of TDP43 are probably playing a major role in several myopathies, such as in inclusion body myopathy. We described a few years ago that TDP43 may also play a role in metabolic disease, at least for the neurological symptoms in Niemann-Pick C, because you have the mislocalization of this protein in the Purkinje neurons that are really one of the major reasons why the patients develop a neuropathology. And so, in a way, TDP43 is not very easy to be defined as a druggable protein, right? Because it's doing so many things, it's playing a role in so many diseases. And so, in my opinion, if we want to look for a cure, or at least a working therapy for ALS, then maybe one of the best strategies would be to find modifiers. And why there should be modifiers? Well, first of all, TDP43 is just the tip of an iceberg, right? There are several proteins that are similar to TDP43 in structure and function, like FAS, HNRPA1, UB1, Matrin3, or TIA1, that have all been suggested to play a role in ALS because there are mutations here that are disease-associated, and this brings to the idea that TDP43 is really the protein that is constituting 97% of the aggregates in the brain, but several other similar proteins could probably cause the same type of disease or affect the TDP43 pathology. And this is something that should not be surprising, because this is a not very recent review on the working of neuronal bodies, axons, and synapses. And you can see that basically every process and every pathway that is going on inside a neuron is controlled by a huge number of RNA binding proteins, which act, I would say, in a combinatorial fashion. And actually, it's only through this combinatorial activity that they can actually allow the neuron to function correctly. And this is something that, another thing that is quite interesting, and in my opinion could help explain a lot of variability that you find in the disease, is the fact that all these HNRP proteins are expressed at different levels in the brain. And this is something that we found, again, a few years ago in collaboration with Leonardo Petruccelli at the Mayo Clinic, where we were investigating the alternative splicing of the SORT1 protein. And SORT1 protein we thought was very important, we know is very important for disease because it's the brain receptor for progranulin. And basically, there was a missplicing event occurring in this, in the SORT1 pre-mRNA, and together with Leo, we became interested in finding what are the proteins that are regulating the missplicing event. And TDP-43 was certainly one of them, but there were also several others. And so, as a part of the work, what we said is, if we look at the level of TDP-43 expression in the neurons, not just of TDP-43, but also of the other proteins that were capable of affecting the inclusion of this exon, what kind of picture do we get? And I think we get a very interesting picture, because what happens is that, of course, for some of the proteins, we could see a difference between healthy patients and patients that were affected by FTLD, so frontotemporal dementia, including TDP-43 bodies. But in my opinion, also a data that was very interesting is that in both healthy patients and also in healthy people, but also in patients, you see that there is quite a huge difference in the level of expression. So you can go from very low levels of expression to very high. And this actually gave us an idea at the time to actually understand, so if all these proteins can participate with TDP-43 in regulating a potentially disease-associated event, and they are all expressed at different levels in people, how do they relate to the TDP-43 pathology? Are they able to compensate for the pathology, or are they able to make it worse? And as Adrian said, this is not something that you can do easily in cell culture. This is, we wanted to have an in vivo readout. You cannot do it with mice because we come from Italy, where research money is very hard to get, and so we would never be able to afford a comprehensive screening of RNA binding proteins. And so we decided to turn to Drosophila, because Drosophila, from this point of view, is a great model. It's a great model for two reasons. The first reason is that if you delete, this is something that we showed a few years ago, if you delete TDP-43 in Drosophila, you get a paralytic phenotype, so something that is a bit similar to ALS. But also Drosophila RNA binding proteins have a very high similarity with human RNA binding proteins, not just with TDP-43. And you can see here in this picture all the major HNRMP binding proteins that have been described several decades ago by Dreyfus and Krejcik and Swanson, that are inside all the cells of our body, and especially neurons. And for all these RNA binding proteins, what we got was a certain number of fly strains in which each one of these proteins, the fly homologue of each one of these proteins, was selectively knocked out using shRNA. And so using this, I would say, a rather comprehensive number of fly strains, what we decided to do is to perform a screening, a functional screening. So basically, we had flies where we knocked out TDP-43, and you can see here they have a paralytic phenotype. We have two different strains, one which knocked it out completely and the other one just partially. And then we also had flies where we actually could see the neurodegeneration in the eye by overexpressing TDP-43. You cannot overexpress it in the whole body because that is lethal. Right? And so basically, we took these three models and we crossed them with the fly strains where we could selectively knock out each of these H-fly HNRMP proteins in turn. And the questions we asked was really very simple. Can we make the flies better, or can we make it worse? Now, I will leave aside the loss of function flies because they were not, in my opinion, very exciting and a little bit disappointingly. But we got some much better results for the gain of function flies. So the ones where the TDP-43 overexpression could give neurodegeneration. Because for these flies, what we got was a very encouraging picture. So we had HNRMP proteins that really were doing nothing at all. So they were not affecting the pathology in the eye. We had some HNRMP proteins that were affecting the pathology, but they made it worse. You can see here, if this is the fly eye that overexpresses TDP43, when you, on this fly eye, you knock out HNRMP U, K, and D, you have an increase in the neuropathology. And then the ones that at the time we were most excited basically were the HNRMPs that could rescue the TDP43 pathology. So if on top of these flies you knock down DASAP1, HNRMPC, HNF, and Q and R, the equivalent of in humans of Q and R, you can see that the fly eye became almost normal. And if you knock them out alone, there was no change. So they were real modifiers of TDP43 pathology. So why do they modify the pathology? Well, I'm going a little bit quickly because this is all published. Basically, the first hypothesis was that they were actually affecting TDP43 localization or expression, but we ruled it out for most of them, but with co-RPA analysis, and we could really see no co-localization, no co-immunoprecipitation. And the only correlation that we could see between these HNRMPs and TDP43 is that they were binding to more or less the same type of transcript. So at the time, we have some very good targets of TDP43 mRNAs in human cells, and if you look at the binding of some of these modifiers, they were binding to this transcript exactly like TDP43. But of course, these transcripts were not very useful to explain the pathology. And therefore, in order to find what we hopefully thought were the transcripts that were actually mediating the pathology, what we did was Rna sequencing analysis. So switching to human cells and neuronal cells, we knocked out, for example, DASAP1, that was the best rescuer of TDP43 pathology. We compared it to the transcriptomic profiles of cells depleted by TDP43. And as you can see here, what you get is really what you expect whenever you work on an RNA binding protein. You have several thousand transcripts that are upregulated and are downregulated. And if you cross these two data sets, what you do get is some information. So, for example, you manage to conclude that both DASAP1 and TDP43 are affecting quite a lot of genes that are having to do with immunity, inflammation, neurodegenerative diseases. This is not surprising, but not really exciting. And also, in general, the well-being of the nervous system. But you can see here that both for downregulated and upregulated genes, what you end up is several hundred transcripts, and you don't really know which ones are important or more important for the pathology and which ones are not. So what we decided to do is to say, okay, if comparing just two data sets is not giving us a lot of information, why don't we compare additional ones? And we decided to concentrate on another TDP43 that could rescue the TDP43 pathology. That was, in flies, it was the SIP protein. But in humans, the SIP protein, through evolution, has actually split and has become HNRMPQ and HNRMPR. And this for us was quite interesting, because what happened is that when we tested and validated Q and R in human cells, what we found is that Q had retained the ability to rescue the TDP43 pathology, but R did not. And the reason is because R cannot shuttle anymore, I think, is because R cannot shuttle anymore from the nucleus to the cytoplasm, but Q has retained this ability that is in common with TDP43. And so we did the transcriptomic analysis of these two cell lines. Again, we found several thousand genes that are upregulated and downregulated. If you look at these two proteins that are really, really very similar from the point of view of the sequence, and you look at the genes that are commonly that are upregulated and downregulated, there is really very little overlap between the two. So that is another, I think, quite important information to give to people who want to work on RNA binding proteins, and the fact that you have two proteins that are really very similar. They come from the same gene evolutionarily, but they really end up regulating very different type of genes. And so now we could narrow down this list of several hundred genes that we have found just comparing TDP43 and DASAP1, because now we have 4 data sets, and we could compare the genes regulated by TDP43, by DASAP1 and HRNPQ that could rescue the pathology, and we also could exclude all the genes that were co-regulated by HRNPR that was completely ineffective in our system. And we ended up with 214 genes, which for me was very interesting, because remember, we were starting from more than 1,000. And these genes were also quite interesting from the point of view of their composition, because these genes had retained the immune-related connections, they had retained the neurological functional connection, that is always quite heartening. There were some connections that before were not so apparent, and these have to do with apoptosis, but again, this is not very exciting. And then, of course, there were just as many that were affecting other pathways. And what for us was also very interesting is that we, in the genes that we collected in this way, we already could see some genes that had already been described using completely different ALS-like models as being protective. So, for example, we found IGF-2 that was described by the laboratory of Stefano Corti as capable of protecting probably the oculomotor functions in ALS patients. And you can see here what happens is that the levels of this protein is not affected by HRNPR, but is downregulated both by TDP43, DASAP1, and HRNPQ. So you could think of a situation where if you have a patient that has TDP43 aggregates and therefore is expressing less of IGF-2, if this patient is producing a low amount of DASAP1 and HRNPQ, then the loss in IGF-2 expression is going to be even higher. Or conversely, if you have high levels of DASAP1 or HRNPQ, then these two can offset the pathology compared by TDP43. And we were all very happy about this, and we decided to sort of ask a grant agency to actually fund the follow-up of this work, right? And say, we have these 214 genes, and some of them are really interesting. Why don't you give us the money to work a little bit more on this? And we almost got the grant. We almost got the grant, except for one reviewer, right, which at the very end said, but you know, 214 is still a very big number, right? I don't know how you are really going to handle that. And so we didn't get it in the end. But I was not really very much disappointed, because what this gave me was the chance to actually improve a little bit our grant application to a different agency, and saying, starting by saying, look, I know very well that 214 is a very big number. So what we're going to do, because in our screening, we had some HNRMP proteins like U, K, and D, which have not been extensively studied until now, and these proteins are capable of worsening the TDP43 pathology. What we're going to do, we're going to actually do RNA seq analysis of cells, neuronal cells depleted by these 3 enhancers of the pathology of TDP43, and then we're going to see, hopefully, what is the kind of overlap that we're going to get with the HNRMPs that can improve improve the TDP43 pathology, and then we're going to test these genes on different ALS disease models, and the hope is to actually end up with just a few genes, or you know, as minimum a number as possible, that you could actually identify as powerful modifiers of TDP43 pathology, and then think of ways of improving their expression and therefore act as a potential therapeutic strategy. So this is something that we have started doing in the lab, and we already have some, I would say, some rather interesting data. The first thing that I think is also very interesting is the fact that if you knock out TDP43 and you look at whether TDP43 can actually affect U, D, or K expression, you don't really see any change. So these proteins are modifiers of TDP43 pathology, but they are not modified by TDP43. And the reverse is also true. If you knock out D, K, and U, and you look at the levels of TDP43 expression, there is really no change. So they are really acting, they are regulated independently in neuronal cells, but they are probably very much acting on the same type of transcript. And so what we have done is to do this analysis. We have performed a very, very deep RNA-seq analysis in order not only to get the information on the gene expression, but at this point we were also interested to get information on the splicing regulation of the that was mediated by these proteins, because there are quite a few reasons why some of these proteins, especially HNPK, as I will show you, could be, become one of the next most interesting players in ALS. And we decided to, as usual, when we did this RNA sequencing experiment, what we got is exactly the same that we got with the other HNPs. So if you knock down HNPD, you have several thousand genes that are upregulated and downregulated to different extent. The same happens for HNPU, you can see here, and also the same happens to HNPK. So looking at these genes singly would not really give us a lot of information. There is still some information that you can gather from this analysis. So, for example, the first information is that if you compare the number of transcripts that are regulated by each of these different genes, they are all different from each other. So they are both acting to worsen TDP43 pathology, but they are not exactly a mimic, a cellular mimic of one another. And this is good because it raises the chance of lowering the number of potential transcripts that are at the base of the pathology. What is interesting is the fact that in some ways they're also very similar, because almost 25% of these transcripts in each protein have to do with neurological and neurodegenerative diseases. The other one are also immune-related and apoptosis-related, although the greatest majority have to do with cancer. So this is something that could, also open a new way. So neurodegeneration and cancer, what are the similarities and what are the differences? But out of these three, the one that for us was very interesting was HNPK, because HNPK, if you look at the effect of depleting HNPK on all the ALS-related genes, that now there is a data bank which numbers more than 120 or so, you can see that they both affect some of the genes that you would expect, but HNPK really is the best one, right? It can affect a lot of, or a higher proportion of ALS-related genes than K and U, and it can also do this at the level of alternative splicing. And you can see that at the level of alternative splicing, although I must confess we still have not validated any of these events, it can actually potentially affect the splicing of CMNR72, Matrin3, progranulin, optinurin, and ataxin-2. And so what we were thinking is that really these genes and these proteins are seeing something that is very similar to what TDP43 is looking at. And also there were some very few reports, but they're coming out, that, for example, HNPK can affect the cytosolic accumulation of TDP43 by controlling CDK2, that is one of the proteins that can apparently phosphorylate TDP43 and cause its mislocalization in the cytoplasm. But for me, the most interesting thing is that if you were to get all these 3 data sets and do exactly the same like we did before, so compare it with the number of genes that is correlated by TDP43, you get down to a number that is very similar to the ones we had before. So we have 54 genes. And in these genes, we could already see some of the proteins that are, some of the factors that are important for ALS. And you could really already see that there was some similarity with the previous data set. And this similarity is, in my opinion, quite intriguing at the moment, because this is really as far as we have got with the experiments. Because you see here, if you take the 214 genes that were in common with TDP43, TAZAP1 and Q, and you compare it with the 54 genes that they are in common with TDP43, D, K, and U, what you end up is with 7 genes. And in these 7 genes, you find again IGF2, that in my opinion now becomes a very interesting target to study. You find also some ubiquitin factors like UBA2E3 that has already been identified by the laboratory of Philip Kale as quite important for TDP43 ubiquitination. TDP43 ubiquitination, you know, that is one of the major hallmark of disease. But also, if you look at all these different other 7 genes, you will see that they are all neuronal, so they have all been described already to have a neuronal function, and some of them are also very, very intriguing, like this nitrogen oxide synthase 1 associated protein that, for example, has already been involved in the formation of dendritic spines at the functional level, but also is become, you know, quite important because of its important role in the exocytotoxic neuronal damage, traumatic nervous system injury, bipolar disorder, and schizophrenia. So we're now currently testing all these genes. The other thing that and this is also something that also includes an advice of when you decide to work on RNA binding proteins, is to always try and validate your results very carefully, because you will get a lot of variability. So in order to actually help narrow down even more the list of genes on which we are going to concentrate, what we did, we got some completely new set of neuronal cell lines from the ATCCs, and instead of the ones that we were using in the lab, that in theory are the same, but in practice they never are. And we did perform the validation on this completely different set of cell lines, and you can see that the validation was pretty good. Out of these 7 genes, almost 4 of them were going in the direction that the RNA-seq analysis indicated. In particular, the nitrogen oxide synthase associated protein is very strongly downregulated, both by depletion of TDP43 and H9NPK. We couldn't test the depletion of all the proteins. We decided to focus just on these 2. And they are also the same target for binding to TDP43, because if you do a immunoprecipitation analysis and you look for the ability of TDP43 and H9NPK to actually pull down the NOS1-AP transcript, you start to get an enrichment in the RNA. So that's a fairly good sign that these proteins are direct binders of TDP43. And what we're going to do now is to actually modulate in primary cortical neurons the expression of TDP43, and all these positive factors or negative factors, will modulate them overexpressing and downregulating them, and see if we can really find a good modifier of TDP43 toxicity that we could then carry on and look for either small molecules or other type of approaches to improve their expression in the brain of in the cells of the patients. So the conclusions of this story basically is that the functionality of TDP43 can be very much modified by RNA binding proteins that are present in the cells, and there are several hundred of them, so we are probably going to find a few more in the future. The fact that if you have common targets can represent a more druggable target than TDP-43 itself, but this is very likely because it's very difficult to upregulate TDP-43 or downregulate it to not be harmful for the cells. The HNRMP, if you do this transcriptomic profile, what you can actually see and get, I think, a first idea of what are the major pathways that are mediated in the neurodegeneration. And I think inflammation is certainly one that will become more and more important in the future. And Bob, I think, is going to speak about this very soon. Some HNRMPs are not like Animal Farm, all animals are equal, all pigs are equal, but some are more equal than others, right? The HNRMPK seems to be particularly involved in regulating ALS genes. In doing this analysis has already allowed us to confirm some of the modifiers that have already been published in the past, but also find some new ones like NOS1AP that is also known as CAPON. And I hope that maybe in the next workshop I will tell you how well these proteins are capable of doing the job of fighting TDP-43 pathology. And this is just to thank the people who did the work. So Maurizio Romano and Straccappelli are the key players in this RNA-seq analysis, and then also all the agencies that are supporting research in Italy. And with this, thank you very much.
Thank you, Emanuele. Oops. It's okay. Thank you, Emanuele, for your amazing presentation again. So, any question for him? You have time for questions? Okay.
Emanuele, I have two quick questions. One, HNRMPK, was that it affects more ALS genes, or does it just affect more genes in general, so that therefore statistically you have more ALS genes affected?
Yeah, we didn't. So if you look at the pathways that are affected by these HNRMP proteins, the percentage of genes in the different pathways is more or less the same, and is higher for HNRMPK and ALS genes. So I would say that yes, the reason why, I don't know, maybe because it has a much better or a similar RNA binding profile to TDP-43, and therefore it's more likely to affect the same type of targets. But this is something that we haven't really got into.
I just wanted to ask one other question, something I've been thinking about recently, which is, do you think that in ALS degeneration always goes through TDP-43? Do you think that's an essential driver of neurodegeneration, or do you think in some cases, or perhaps other genetic cases, you can have neurodegeneration without going through a TDP pathway? What do you think?
No, this is a very nice hypothesis. I would say that these data tell you really that you could have different pathways that could be involved, and not necessarily TDP-43 maybe the beginning of the pathological cascade, right? Then eventually you're going to, because 97% of the ALS patients will have TDP-43 aggregates, but not necessarily this has to be the first, right, to go wrong. You could get it later. Yeah.
I just want to comment on the last phrase of all. Be careful, Bob sitting next to me. So it's well described. People always say this. Everyone with sporadic ALS has TDP-43 aggregates, and then people show pictures of those mislocalization that you see. In reality, pathologically, it's exceptionally rare. It's not a common pathologic observation. You talk to neuropathologists and say, sure, we can find it. gotta look really hard to find it in a lot of brains. Some it's very prominent, but it's not like the entire brain has that. There can be little areas that have some, and most common is the cytoplasm that's localization, not always the aggregates.
Right.
But even then, it's uncommon. And I only say that because it doesn't mean that everyone with sporadic ALS with TDP43 is the upstream mechanism. That's a hypothesis. There's not good data yet that supports it, other than that limited pathology. You want to say something?
I do. I mean, you know, it's tough because a lot of cases you don't know where they're looking, right? So TB tends to be in the areas of active generation, definitely number one. But the case series are, you know, less than 5% of neurons in an area where people look. It's sort of the better way to think of it. And that you're totally right about that. It's often there, but it's not always the most prominent. I mean, I think the interesting thing on that note is the C9 cases that have been observed prior to getting neurodegeneration. In each of those cases, they had DPRs, they had foci, but they didn't have TDP. And so it seems, and until we get a PET ligand or something, we won't know, but it happens right around the time of degeneration. And who knows if that's a good target, bad or otherwise, but it's not bad.
I have a real question. No, seriously. So you're talking about your HNRMPs, and what intrigued me is that we see the same HNRMPs that also modify toxicity induced by hexanucleotide repeats, as you might know. And in that context, I was wondering, in your flies, do you see an improvement if you overexpress these HNRMPs? Because now you just downregulated them. So
Yeah.
it would be very interesting to know what happens if you overexpress them.
Yeah, I totally agree. We just didn't get around to do this because we wanted to move on the human side. And the other problem, you know, there is also a little bit of I think of a technical problem here with the overexpression, because these HNRMPs tend to be very highly expressed, right? So what happens, for example, I have more experience for TDP43, but you might imagine is in the same situation as the other HNRMPs. So for proteins that are very highly expressed, overexpressing them to a level that you will be able to see a functional readout can be, I would say, pretty challenging, right? And what is very likely is that they are going to be toxic, but because you are going way beyond any physiological meaning, right? Downregulation in these cases, I would say, is more, you get results that are more reliable, right? But I can tell, so for example, just to tell you an example, you know where you can see, so you know that one of the original target of TDP43 that I found in 2001 is CFTR exon 9. Nothing to do with ALS or anything like that. But it has to do with cystic fibrosis and congenital blood. So there is another protein. So if you overexpress TDP43 into cells, you don't really see a big difference when you look for CFTR exon 9 splicing, because there is already so much. It's very difficult to go beyond the threshold, right? On the other hand, there are several proteins, HNRMP proteins, that are binding to UG repeats, like TDP43. So, for example, ETR1, CUGBP, right? And these proteins are very lowly expressed in cells. So when you overexpress ETR1 will actually compete with TDP43 for the CFTR exon 9 splicing event, and then you will see very clearly the effect of the overexpression. But you have to start from low level.
But could it be that in pathological situations you have not a sufficient amount of these RNA binding proteins, and that by overexpressing it you can make a difference, and that could be from a therapeutic point of view could be very interesting.
Yeah, that is possible. But you can see from the test data, I mean, one of the data I like the most from our collaboration with Len is the fact that you can have 4, 5, 6-fold differences in different people. Right? And this can also vary over time, because they're not stable. Right? TDP-43 levels apparently decrease with age. So we have a very dynamic situation. And that is, I think, probably why we have this huge phenotypic variability, because you have many players.
More questions for Emmanuel? No more? Okay, thank you again, Emmanuel. And now we have 30 minutes for a coffee break, so see you again here at 5:30. Yeah. Okay, good afternoon again. We continue with the second session of this workshop. The next speaker is Caroline Rouen. She obtained her PhD from the University of Strasbourg in France, where she studied epigenetic mechanisms that accompany neuronal apoptosis and neurodegeneration. There, she started working on ALS mouse models and test pharmacological approaches to restore epigenetic equilibrium in motor neurons. After defending her PhD, she joined the lab of Paola Arlota at Harvard University, where she studied the molecular mechanisms that govern the specification of cortical neuronal populations. This allowed her to develop programming and reprogramming strategies in vivo. In 2013, she returned to Strasbourg as an assistant professor in the research unit of Luc Dupuy and developed her own research program that interrogates the contribution of the cerebral cortex and its neuronal populations to ALS onsets and progression using mouse models of the disease. So, Caroline, when you want. Thank you.
Thank you very much for this introduction, and I also would like to thank the organizers for offering me the opportunity to present our work today. So I will not talk about new genes or new treatment, but hopefully new hopes. And I would like to talk more particularly about different cell types, which are the corticospinal neurons. So I have decided to give a relatively short introduction because all of you know probably more than I about amyotrophic lateral sclerosis. And I would like to concentrate mostly on unpublished work. So my introduction is the next slide. You all know that amyotrophic lateral sclerosis is defined in clinics and in terms of pathology as well as the degeneration of two neuronal populations, the corticospinal neurons in the cerebral cortex and the spinal, bulbar and spinal motor neurons in the core. And so in clinics, signs for the degeneration of these two populations arise as weakness, atrophy, and fasciculation as it comes to spinal motor neuron degeneration, and spasticity, slowness of motion, hyperreflexia, and hyperexcitability of the motor cortex as it comes to upper motor neuron or corticospinal neuron degeneration. And so if you want to translate that into preclinical research, then you end up thinking that all of those signs are extremely difficult to find in a worm, a Drosophila, but also in a mouse. And so that may be one of the reasons why there is a big discrepancy between clinical presentation of ALS and preclinical research on animal models, since we now know a lot about the different mechanisms that are involved in lower motor neuron degeneration, but we still don't know how corticospinal neuron degenerates. And so this in spite of the fact that even mouse models have already been shown a while ago to recapitulate the loss of corticospinal motor neurons. This was shown initially by Zhang and Shima in 2002, and this was followed some 10 years later by Jeff McLeod and Hande Hosiner when she was a postdoc in his lab, where they showed a progressive loss of corticospinal neurons, and which started already at presymptomatic ages. And we also know, thanks to the work done by Thompson in the lab of Clive Benson, that the cortex contributes to disease onset and progression. And this was shown by reducing the expression of SOD in the cerebral cortex of G93A rats. So all of these suggest that rodent models should nevertheless be good models to study amyotrophic lateral sclerosis, and more particularly, the contribution of the cortex and of the corticospinal neurons to the disease. And so this is the questions that we have tackled some years ago when I went back to France. And the question was to understand the role of the cerebral cortex and of the corticospinal neurons in ALS onset and progression. And then we started with asking two questions. The first one is, how do corticospinal neurons contribute to disease onset and progression? And the second is, what are the molecular mechanisms behind corticospinal neuron death and dysfunction during the course of the disease? And so the work I will present today was performed by Thibaut Bird, who recently graduated. Elena Sekic-Zajerovic, a postdoc in the lab, and Christine Marquez, who did her PhD with me and who is now a postdoc with Brian Wanger at MGH and Harvard Medical School. So just one slide to present the mouse cerebral cortex in a nutshell. You all know that the cerebral cortex is organized in layers. It's a very well-organized structure composed of 6 layers where you can find specific subtypes of excitatory glutamatergic neurons, inhibitory GABAergic neurons, and a bunch of glia, which are probably very diverse as well, but that we may not know very well yet. And so if we concentrate on the excitatory neurons, they arise in, they can be classified in different subtypes according to the way they project their axons. The layer 6 here in green is composed of corticothalamic neurons that send their axons to the thalamus. Layer 5 in yellow is composed of subcerebral projection neurons, which are the main output of the cerebral cortex, and that comprise, among others, the corticospinal neurons. So they send axons, among others, to the pons and the spinal cord. And the upper layers, but also the other layers, are composed of callosal projection neurons, which connect the 2 hemispheres of the cerebral cortex together. So I will talk about the subcerebral projection neurons and mostly corticospinal neurons, but also about callosal projection neurons in my talk today. So to tackle these questions, we started with a well-known mouse model of the disease, the SOD1 G86R mice, which are similar to the SOD1 G93A with a few differences, but we used them because we knew that at least they recapitulated cardinal features of ALS. And SOD1 mice at the time also came as a conditional models, which was also useful for the questions we were asking. So these animals, they start showing denervation of the neuromuscular junction and motor symptoms at around 90 days of age, and they die pretty fast after this, within only 2 weeks to 20 days. And my colleague Luc Dupuis previously showed that before this, weight loss was already observed in these animals along in line with the metabolic hypothesis in ALS. So using this model, the first thing that we wondered was whether these animals also recapitulated corticospinal neuron degeneration, and I will try to go fast on this because this is not extremely original. So what we did, like other before us, we performed retrograde labeling of this neuronal population, which cannot be observed by any kind of staining. The best that we can do is to label the whole layer 5 of the cerebral cortex that includes corticospinal neurons, but many other excitatory projection neurons as well. So here we perform retrograde labeling, and then we sample the whole population along the rostrocaudal axis of the cerebral cortex and quantify those neurons. And so we observed a loss, a progressive loss, or progressive disappearance of our marker during the course of the disease, and quantification indicated that we had a significant loss starting at 90 days of age, which matches with the beginning of the motor symptoms, and that increases over time. But because we sampled the whole rostrocaudal axis, we wondered whether there were more prominent effects at certain parts of the brain. And at 90 days, when we start seeing a significant decrease of the population, we observed that the sections that were the most affected were the most caudally located ones. And so it seems that over time, this degeneration progresses from the most caudal to the most rostral part of the cortex. And for us, this was very interesting because it was already described that the most caudal part of the motor area is composed of the corticospinal neurons that send their axon to the lumbar part of the spinal cord. And in the SOD1 mouse, it is very well known that the motor symptoms start in the hind limbs that are innervated by the spinal motor neurons. So this suggested that there may be a somatotopic relationship between the corticospinal neuron degeneration and the spinal motor neuron degeneration, as it had been reported in patients before. And to check this, we performed a second series of retrograde labeling, this time from the lumbar spinal cord, and as you can appreciate here, we label much less corticospinal neurons, mostly those located in the most caudal section. And when we quantified them, we observed that the loss of this population was already present as soon as 60 days of age, this time which is much earlier before symptom onset. So, as I told you before, in those animals, motor symptoms arise only at 90 days of age, and so while it was already published, we verified this on the very same animals. And so we observed that the denervation of the muscles, as assessed by EMG here, and neuromuscular junction denervation, as assessed by immunofluorescence here, were evident only from 90 days on. So if I summarize this first part, in these G86R animals, denervation starts at 90 days, weight loss occurs before, but corticospinal neuron loss starts much earlier, and it starts mostly with those corticospinal neurons that project to the lumbar part of the spinal cord. And I didn't show it here, but in those animals, the loss of the cell bodies of the motor neurons is extremely late. So we had a good model to assess the contribution of these neurons to ALS onset and progression. And so we asked two sub-questions. The first one is, would amyotrophic lateral sclerosis, or at least its symptoms, or what its features in mice, ever develop in absence of corticospinal neurons? And that would be one way to test for their contribution, but that also would be going against the definition of the disease, since we wouldn't have the loss of corticospinal neurons. So to test this possibility, we generated a mouse that overexpressed SOD1 G86R ubiquitously, but that totally lacked the corticospinal neurons. And to do so, we crossed the G86R mice with the FETF2 knockout mice, which were generated by Hirata and collaborators in 2004, and that the Mac-Lislav further phenotyped, and they described that these animals completely lost all the subcerebral projection neurons, which are labeled here by CTIP2. So all the neurons, the excitatory projection neurons of the layer 5 of the cerebral cortex. And so accordingly, without those neurons, there is no more connection between the cerebral cortex and the spinal cord, as shown here. This is close up on the sagittal section with an anterograde labeling from the cerebral cortex. So you still have connections to the thalamus by layer 6 neurons, but no more connections to the spinal cord. So with these animals, we generated, so we crossed the FETF2 mice with the SOD1 and generated 4 genotypes of interest. The FETF2 wild-type animals, non-transgenic, which are simply called wild-type. The FETF2 knockout mice, non-transgenic, which are simply called knockouts. Mice, wild-type of FETF2, and that overexpresses SOD1 G86R transgene, which I will only call SOD1, and the double mutant knockout for FETF2 and overexpressing SOD1. And so in control experiments, we verified that in absence of FETF2, so in the knockout and knockout cells, we did not have any more layer 5 neurons labeled here by CTIP2, and that the absence of FETF2, as assessed here by qPCR, did not affect the expression of the SOD1 transgene neither in the cortex nor in the cord, and it did not affect neither expression of CAT in the spinal cord, which we use here just as a marker of generation and specification of the motor neurons. But I should say that FETF2 is not expressed in the cord, so we were not expecting any phenotype there. And so here are the results that we obtained. So the first thing here is that our animals were smaller. The knockout and knockout third one were smaller. This is weight at the beginning of the study that were smaller than the wild type and the third animals. And we were worried that this may be an issue for them because As we discussed already this morning, metabolism is important, and so we were worried that having thinner and smaller animals may be detrimental to their survival. So they are smaller, but the knockout animals for SLF2, they normally gain weight over time. As already reported, the SLD1 animals quickly stop losing weight and they quickly stop gaining weight, and they stop losing weight. But the double transgenic, as you can see here, or the double mutant, they also stop losing weight, gaining weight, but they don't lose so much. And if we use here weight loss at the onset of the disease, we observe a delay of disease onset in the double mutant animals, and a delay also in the survival. And so altogether, this suggests that absence of corticospinal neurons is rather beneficial for the disease, since it delays onset and it extends survival. The disease duration instead was not affected. It was similar in both cases, but the weight loss over the course of the disease was much smaller in the double mutant animals, which was also very surprising to us because, as I said before, and as it was mentioned this morning, the weight loss is also a good indicator of disease progression. And so this is a fact that this is something that we cannot explain at the moment, but it goes together well with the increased survival. So I will now move on to a second sub-question. The second sub-question that we asked is, would ALS ever develop with wild-type-like corticospinal neurons? And to answer this question, we used this time the SOD1 G37R mice developed in the Complutense lab that have a flux transgene which can be removed upon recombination by CRE. And we tested them the effect of the removal of this transgene selectively from layer 5 subcerebral projection neurons. And so to do so, we generated a mouse line that expresses the CRE, an inducible form of the CRE recombinase, under the promoter of CRE. The construct is here, so it is a knock-in. And we choose CRE because it is nicely expressed in the layer 5, so not only the corticospinal neurons, but all the subcerebral projection neurons. And it is not expressed in the spinal cord nor in the muscle. It is also expressed, I should add, in the most dorsal and medial part of the cerebellum. So as you can see here, these are in situ hybridization and the immunofluorescence that shows the nice expression of CRE in layer 5. And so we generated those CRE CREAT2 mice and we crossed them with the TD tomato to check for recombination. And we observed that TD tomato expression could nicely recapitulate that of CRE with nice expression in the layer 5. And when we retrogradely labeled our corticospinal neurons, we could also see colocalization. So again, here it's not only corticospinal neuron, it's the whole layer 5 subcerebral projection neurons. Importantly, in the spinal cord here, you see the dorsal funiculus nicely labeled by TD tomato, located in the axons of the descending corticospinal tract, like CRE. But importantly, there is no recombination in the spinal motor neurons or other neuronal cell types of the core. So with these animals, we performed, we first verified whether CRE mice could excite the SOD1 transgene from the subcerebral projection neurons located in the layer 5. And so we microdissected layer 5 and layer 2-3 as control, and we observed the decreased expression of SOD1 here. And we were not expecting a full disappearance of SOD1 expression, because here we microdissect the layer 5 that contains the subcerebral projection neurons where the recombination occurs, but also interneurons and different types of glia. So this is the decrease here is significant, but we were not expecting a full disappearance. And then in control layer 2-3, as you see, SOD1 remains highly overexpressed. And so we retrogradely labeled the corticospinal neurons and performed quantification. And what we observed is that the removal of this transgene from the corticospinal neurons was sufficient to promote the survival of this population. In other words, this manipulation was neuroprotective, and the way we can interpret this is that the loss of corticospinal neurons is very likely depending on cell intrinsic mechanisms, because removal of SOD1 is sufficient to promote their survival. This was accompanied by decreased expression of spasticity. So what we did here is that we performed a measurement of the long-lasting reflex in the tail of end-stage animals. So they need to be fully paralyzed to run the EMG test. And so what we observed is that the occurrence of long-lasting reflex was decreased in the cream flock SOD animals, so upon removal of SOD1 in layer 5 neurons, as opposed to the control SOD animals. And for those that remained, that still displayed the long-lasting reflex, its amplitude was also decreased. So altogether, this goes together with promoted survival of corticospinal neurons and probably promoted increased function as well. But in this condition, we were very disappointed to see that disease onset of late stage of the disease and survival was totally unaffected. So in other words, if I summarize these two sets of data, absence of corticospinal neurons in the SOD1 mouse is beneficial since it delays disease onset and increases survival. But removal of SOD1 transgene within the corticospinal neurons and maintenance of this population does not promote improvement in terms of onset of survival, even though it protects the corticospinal neurons themselves and induces a decreased spasticity. So with this, I will move to the last part of my talk, which has been dedicated to the investigation of the molecular mechanisms behind the dysfunction and the loss of corticospinal neurons during ALS. So as I mentioned, this is the work on 2 markers. And so what we did here was first to try to find a way to purify those corticospinal neurons from the cerebral cortex of the animals in order to really decipher what was going on in these specific cell types as opposed to the whole cerebral cortex, since they are the one that shows degeneration. So in order to label those neurons, again, we performed retrograde labeling. This time we used a different marker. And then at age of interest, we sectioned the brain, microdissected the area, the layer 5, and then performed fast purification. And along the way, we took a control population, the callosal projection neurons of the upper layers, which I mentioned previously, that are very closely related excitatory projection neurons of the cerebral cortex, but that are located in the upper layers, and importantly, that do not degenerate in ALS, and we verified that they also do not degenerate in our mouse model. So this is our control population, and the idea here was to be able to separate the transcriptomic dysregulation, which was simply due to SOD1 overexpression, and which we assume we would see in both neuronal populations, from those that are really involved in dysfunction and degeneration, and that we assume we would see only in the corticospinal neurons. So upon purification of these 2 cell types, we ran RNA-seq analysis, and we did this at 4 different time points, 30 and 60 days of age, which are pre-symptomatic ages in those animals, and then 90 and 105 days of age, which are symptomatic ages. And I would like just to remind you that at 60 days, we already observed loss of corticospinal neurons, only those that are located in the most coral part of the brain. So fast purification of these 2 populations was extremely difficult. It took us quite a lot of time, but we were happy eventually to collect enough neurons to perform our RNA-seq analysis. And so non-supervised hierarchical clustering of our samples, nicely separated here the corticospinal neurons from transgenic and wild-type animals. from the callosal projection neurons. And when we verified the expression of genes known to be expressed in either one or the other population, we could see what we were expecting, markers of corticospinal neurons upregulated in this population than regulated in the callosal projection neuron, and vice versa. So we started with principal component analysis, and I would like to show you first the right panel here, which corresponds to the callosal projection neurons. Here there is a complicated color code, but what I would like to say is that basically we were not able to discriminate wild type from transgenic animals. And this matches with the fact that we know that those neurons do not degenerate in ALS. On the other side, sorry, and here we remove the third one gene to really see what's going on besides third one overexpression. On the other hand, for the corticospinal neurons, I draw a dotted line here. It's not a clear cut, but basically on the left part you have the warm color, red, orange, and yellow, corresponding to the mutant animals, and on the right part you have the green colors corresponding to the wild type, and so we could discriminate between the two genotypes by PCA analysis. And so we looked at those 4 individual time points, and what we observed in the gray circles here is that there was not many difference going on in the callosal projection neurons over time, while there were bigger transcriptomic alterations taking place in the corticospinal neurons over time, and as you can see here, there is a peak at 90 days of genes. Then after this, we ran gene set enrichment analysis to try to understand what were the main pathways going on in molecular pathways involved in corticospinal neuron dysfunction and degeneration. And I don't know whether you can read here, but what we observed is that there were several pathways involved in RNA metabolism, such as here ribonucleic protein complex biogenesis and assembly, translational initiation, RNA processing, tRNA metabolic process. And so we were quite surprised to see this in a model overexpressing SOD1. So of course we observed the mitochondria was the first geotem to come up, but that was not much of a surprise. But this here was more surprising, and so we thought that maybe here we could have a link with what was already known from other genes like first TDP43 or even C9. And so we wonder whether it's by going at the individual neuronal population level that we may see those dysregulation upon SOD1 overexpression. And so this suggested to us that RNA metabolism was altered, and so when we looked at some of these genes, we could see that some, here you have wild type on the wild type samples on the left side, such samples on the right side, and you see the upregulation of most of these genes. And so here I have color-coded some of them. In red, we observe genes that were linked to a hereditary spastic paraplegia, primary lateral sclerosis, Charcot-Marie tooth disease, and spasticity. So that was not very surprising, but we were happy to find them anyway. Then in purple, there were genes related to spinal muscular atrophy, so we are still in the motor neuron disease field. And then in blue, genes linked to X-linked hereditary ataxia, and then some other genes linked to mental retardation. So with this last category, we go maybe more specifically to the cerebral cortex. And so because we saw so many alterations of RNA metabolism, we wondered whether splicing was altered in our samples, and we had a sequencing that was deep enough to allow us to also interrogate this. And so we could qualify several types of alternative splicing, like skipped exon, alternative 5' splice sites, alternative 3' splice sites, mutually exclusive exons, and retained introns. And so when we performed gene set enrichment analysis for all those genes that showed altered splicing, we find here genes involved in synaptic signaling, regulation of neuronal differentiation, death. And so here we were really into the function of the neurons. And so you can see some examples here of altered splicing. So with this, I will conclude this last part. So we have developed a method to purify the corticospinal and the callosal projection neurons from the cortex of transgenic mice, and we ran a Rna Seq analysis, and we observed alteration that were selective to the corticospinal neurons compared to the control callosal projection neurons. We came up with altered RNA metabolism, which points to common mechanism in amyotrophic lateral sclerosis. And then we observed after splicing that particularly affects genes involved in neuronal development, but also synaptic transmission. And so I will summarize here the whole talk. So I've shown you that SOD1-G86 R mice recapitulate corticospinal degeneration, and they do so prior to motor neuron degeneration. We also observed a somatotopic relationship between the loss of corticospinal neurons and that of spinal motor neurons, similar to what has been already reported in ALS patients. Then, when we manipulated this population, we observed that the absence of the corticospinal neurons and other subcerebral projection neurons of the cortical layer 5 delayed disease onset and extended survival, suggesting that in the mouse model of ALS, corticospinal neurons may be somehow toxic. And so excision of the mutant transgene SOD1-G37R from this population and other subcerebral projection neurons is sufficient to promote the survival of this population, but does not affect neither onset nor the survival. So suggesting that it's better, if you are a mouse model of ALS, it's better to have to not have corticospinal neurons than to have wild-type like corticospinal neurons. And then we developed a method to purify neuronal population from the mouse cerebral cortex under both healthy and neurodegenerative conditions, and ran longitudinal transcriptomic analysis, which allowed us to develop a comprehensive analysis of transcriptional alteration that takes place in the corticospinal neurons as they degenerate in the third mice. And that pointed to two mechanisms. One is altered RNA metabolism, as I said before, which is reminiscent of other genes known to be involved in ALS, but also developmental and synaptic alterations, which point here to the proper functioning of the neurons. And so with this, I would like to thank the people who did the work, Christine Marquez, now as I said, a postdoc at NGH, and Harvard Medical School, Matthew Fisher, who helped her a lot. He was a technician in the lab and he now moved to Oxford. Rna Sequencing postdoc, and then Thibaut Burke, who just graduated. This is the rest of our intern unit in Strasbourg, and these are the agencies that funded this work. And with this, I would like to thank you for your attention, and I will be happy to take any questions.
Thank you, Caroline, for your great presentation. Any questions from here?
Thanks. That was really nice. That was a beautiful dissection of the biology of the upper motor neuron. One thing that you didn't look at, I'll suggest, and though you got a hint from your transcriptomic analysis, is when Hyundai first published those mice, one of the key findings, which matches mouse and human, is dendritic simplification, the loss of dendritic spines. And with a normal upper motor neuron that you're showing, you didn't change the disease course, but you actually didn't look to see if the surrounding milieu, meaning the glia, if you will, are affecting that functional property. You get a hint from your transcriptomic on synaptic biology. Now, the reason I also ask this, so I'm going to suggest you actually look at those mice in dendritic spines, because we discovered that they're layer-specific astrocytes, layer 2 to 5, that regulate spine development. And if those astrocytes are dysfunctional, you'll lose spines. So if your cells, because of the mutant SOD1, or you pick whatever the toxin is, they lose those spines, they're going to have not the same effect on lower, whatever that biology is on lower motor neurons. So I'm going to suggest you look at that.
Yes, thank you. Well, I totally agree with you with the role of the milieu, and this is really what the excision experiment showed us, is that if you still have like wild-type like corticospinal neurons within a mutant environment, the disease occurs nevertheless, and you do not affect neither the onset nor the survival. So for sure, the surrounding milieu or environment has a very important role. And so whether these are various glial cell type is one possibility. We were also thinking of interneurons because these also have been shown to play a role. In our mouse, we haven't looked, I mean, we did some quantification of interneurons and didn't see any difference, but we also ran some qPCR analysis for various glial markers, and we did a few immuno as well. But in the cortex of those animals, we saw no difference. So,
we'll talk later.
Yeah.
And this is not about exercise killing. That's a lot of in vitro stuff. There's no good data in vivo. This is about modulation by the synapse. There's a growing literature on that.
Yes. But this is very, as you say, this is very possible given that we see so many synaptic genes involved in the dysfunction of the cells. Thank you.
I have a question concerning the projection. Do you also see not only corticospinal projection, but do you also see a corticothalamic projection? And the question is, do you see in your mice also a decrease of neurons in the thalamus? Do you see any change there? Is just following the spreading along lines?
We haven't checked neither the thalamus nor the corticothalamic neurons in the layer 6. I think that if there is a loss, it must be discrete and it requires intensive quantification like we did in layer 5. So I cannot answer at the moment, but it's possible, as you say, given the possible progression of the disease. Yeah.
Thanks, nice work. I wonder if you could comment, considering the huge functional difference that there are between corticospinal neurons in mice, rats, to humans. Because as you indicated indirectly, a mouse without cortex may live, may walk, may feed, may have sex, everything. But the human has a lot of impairment, so probably we are talking about a different segregation of function. So could you comment, or could be useful your information to be compared with, for example, array or certain analysis on human neurons to see?
I totally agree. I mean, the differences are so important that it may also be one of the reasons why we didn't look so much at the cerebral cortex and at the corticospinal neurons in the first place. But what is striking to me is not the difference, it's the similarities. I mean, we here have a mouse model that, as you say, can very well live without corticospinal neurons. Yet, their corticospinal neurons degenerate during the course of the disease. And so I think this is very remarkable. What is also remarkable, and that I didn't highlight here, is that in patients, from what I understood, so the one mutant carrier have very little upper motor neuron signs. And so it is possible that we here have a model where corticospinal neuron is rather small. It's only 30% by disease end stage, but maybe that would be much more important in other mouse models, and I think that we should look into this as well. So I agree with you, there are big differences, but I think we should concentrate on what is common if we want to dig more into this. And the last reason I would like to give is that as opposed to motor neurons, we still do not have a good in vitro or culture model for those neurons. I mean, we are not able to generate from iPS corticospinal neurons, and so as of today, we have to learn with the tools that we have, and we can hope that they will get better over time.
I can shout if you want.
I would like to ask you a question about the survival curve, where you have removed the corticospinal neurons. It was clear that there were two populations. Do you have an explanation for that?
Yes, absolutely. These are a mixed background. The SOD1 GAC6 are mice are FBB mice, and FEDF2 knockout are on the C57 background. And because we did two rounds of crossing, what we observed was already reported in other mixed backgrounds, so we were not surprised, and we were expecting this actually.
This is not a female-male difference?
No, no.
And can that not influence your results? Because I'm always afraid of mixed backgrounds.
Actually, we only had males. I don't know how females would survive. We did everything on males. Yeah.
Yeah, but then I come back to my question. So, are you not afraid that the mixed background has an effect on what you see? And you can argue that both have a mixed background.
I mean, there is definitely an effect because the curve not only is in two phases, as you mentioned, but also the mice die much later. I mean, the effect is there and it's obvious. Because it was already reported, we were not too worried. And then we controlled for it by comparing with simple transgenic mice, which were also on a mixed background. There were little males.
It was different to make a distinction between the effect of removing the corticospinal neurons and the mixed background. You're comparing two different things at the same time.
Yeah, but the simple SOD1 mice, which we call just SOD1, were also on mixed background. There were very little males.
They can be differently mixed.
Oh, okay.
Okay.
Thank you.
No more questions for Karen? Thank you again so much.